Hi, this is our lesson 5 in Unit 4 Molecular Genetics, and we're going to look at the different types of mutations. So before we do that, I kind of want to revise on how protein folds and why the sequence of amino acid is so important, and therefore you need to have no errors in your DNA. So if we look at the protein structure, right here you have a chain of amino acid. And if we zoom into one of these amino acid, let's take one of them and zoom in on here and look at the chemical structure. There are three major components. There is number one, the carboxyl group on here. And then there is the amino group on the left-hand side. And here represented by an R, that is called the side chain or the R group, or you can also call it the functional group. So there are three names for this thing. Now, this has been taught in Unit 1 and also earlier in this unit. The amino acid itself, the amino group and the carboxyl group on all amino acids, they are the same. They're identical. So the chemical structure of these two bits, the green and the purple bit, doesn't change. The only part that actually varies would be the functional group, the R chain, which is shown by the highlighted boxes right here. And these causes them to have different properties. So each amino acid has its own property, and those properties could be polar, as you can see in the blue group. They could be non-polar, they could be positively charged, and they can also be negatively charged. Again, this was taught in Unit 1, so if you don't remember, you can go back and look at those videos again and see how they folded. If you're too lazy, I've also got a really good YouTube video by the Amoeba Sisters. They're amazing. And you can play it at home and see what the structure of protein is and how they fold. And that's going to help you understand why DNA sequence is so important and how that leads to amino acid sequence and how that amino acid sequence leads to the shape of a protein and how it folds and how that shape is highly related to a protein's function. So I'm not going to play it in my video. You can look at it at home. Next, we have the definition of mutation. So mutations in biology talks about any changes that happens to the genetic material. And these mutations can be at a very grand scale where you have changes to the chromosome, or you can have it at a very small scale when you're changing only one single base in your genetic material. So as long as there is a change, it is called a mutation. There are different ways of categorizing mutation when we ask the question, oh, what type of mutation is this? So the first way to classify it is based on the effect it has on the organism, how it is affecting the organism that has the mutation. So these effects are categorized into beneficial, neutral, and deleterious. So if we say that a mutation is beneficial, it means that the mutation increases the organism's chance of survival or reproduction. If we say that it's neutral, it doesn't really affect the chance of the organism's survival or reproduction. If it is deleterious, means that it's deleting something, then the mutation actually decreases the chance of survival and reproduction. So I think this in itself is not hard to understand. However, do understand that whether a mutation is beneficial or deleterious highly depends on the environment that the organism is in. So if we think about the human population, um, the skin color, for example, let's say everybody had a medium skin color, like yellow, right? Like Asians. And then suddenly somebody had a mutation and their skin became darker. Now, this can be beneficial mutation if that person's location is in Africa, in a place where it's super hot and super sunny. It would help protect the person from skin cancer. However, it is not a beneficial mutation if that person locates in the North Pole, for example, where there's just snow everywhere and everything is white, and there could be predators roaming around trying to eat you, and you really stand out from that white background. So, whether or not a mutation is beneficial or deleterious or neutral really depends on the situation itself and the environment of that organism, where that organism is in. So, after the effect of mutation, we also look at the mechanism of mutation, or you can call it the scale of that mutation. So first, we're going to look at gene mutation, where the mutation happens to only one single gene in that long strand of DNA. Remember, your DNA is super long and it has multiple genes on it. So if the mutation only affects one single gene, then we call it a gene mutation. If the mutation affects the entire chromosome, which is the entire strand of DNA, or multiple strands of DNA, which is multiple chromosomes, then we call it a chromosomal mutation here at the bottom. So let's look at the first one, gene mutation. So a gene mutation, usually it's also called a point mutation because you have mutation at one single point. And that is this very small scale mutation. So usually it happens when you have a change in one single base. So there are three types of point mutation. You can see that there's substitution, insertion, and then also deletion. Sorry, my nose is quite blocked at the moment. So substitution reaction is when you are substituting one base. So if we look at this diagram right here, this is the normal DNA, and this is the normal mRNA produced from that normal DNA, and this is the resulting amino acids. And remember, the ultimate goal of having DNA is so that you can produce a correct sequence of amino acid. So when we look at mutation, and the effect and the scale, we usually refer to the amino acid, the final result, and what we care about most, and see how the amino acids that get produced is affected. So substitution is when you substitute one single base, and you can see in this case, this G has become an A. So if it has become an A, then you can see the resulting amino acids that this codon is coding for. If you don't remember the word codon, you really need to revise lesson 2 in this unit. So this codon codes for a specific amino acid, and you can see that it has been changed from GLU to LYS. So it's got changed to lysine. So that's what substitution mutation is. Now, the next one is called insertion. So insertion is when you are inserting one base into the original sequence. So for example, right here, you can see that in front of this G, another G has been inserted. So after inserting this G, when the ribosome is reading the mRNA, it is still reading codons, which means these mRNA bases are being read in triplets. And so you can see that this one goes along and then it has produced what we call a frame shift effect. Everything downstream from that insertion is wrong. You can see this amino acid has been changed. This amino acid has also been changed. And then next we have deletion. Deletion is just like insertion. Instead of inserting a base, you are deleting a base. So we are deleting a base right here. So originally we had a U and then an A and a C. And you can see that we're deleting that U, and then it becomes just AC, and the following G belongs to the second codon. And this also produced a frame shift effect, but in that opposite direction. So everything downstream is wrong after that point. It's just like when you're doing a multiple choice on an answer sheet and you missed one question and then you just filled up the space in front of it. And so all your answers after that missed spot became wrong because you're filling in your answers at the wrong place. So it's just like that. So which of these point mutation has the least effect on the amino acid chain produced? Well, substitution, because you're substituting one base. And so technically speaking, only one amino acid is affected. If you have insertion or deletion due to the frame shift effect, everything downstream of that point of mutation became wrong after that. Okay, so I have to look at what the mechanisms of mutations are. You can also look at the outcome of the amino acid and categorize it using the outcome. So for point mutations, where you're only substituting a base, you can actually produce these four different types of mutation. Those mutation can be silent, it can be nonsense, it could be conservative missense mutation. It can also be non-conservative missense mutation. So let's look at this normal one first. So this is where you have no mutation at all. And when you have no mutation, you can see that lysine is being produced. And if we look at the silent mutation, even though you've changed one of the base, you can see that this base still becomes this codon, and then this codon still code for the same amino acid sequence. So meaning that you changed the base, but you haven't changed the amino acid it's coding for. And therefore we call it silent because you're not going to notice it at all. It still has the same amino acid, meaning that the chain will still fold in the same way, and that's not going to affect the shape of the protein, nor is it going to affect the function of the protein. Now, a nonsense mutation is when you have a change in the base, and that's going to result in a stop codon. So if you look up UAG in the codon table on the code or the codon wheel, you'll notice that it is coding for a stop codon. And that stop codon, when you do your translation, it's going to cause a release factor to come in, and it's going to put a stop to your translation and to your protein production. Meaning that when you hit this codon, your protein building is going to stop right there. So you get a half-produced protein, half-done protein. Missense, if we look at this, is when you have different amino acids produced. So you can see for both of these sequences, we had a mutation, and the protein produced from that is arginine or thriosine, however you want to pronounce it. However, for the missense mutation, there are two types. One is called conservative, the other one's called non-conservative. And this really depends on the property of the amino acid. So let's compare this one, the conservative, with the no mutation one. So no mutation is lysine, and then after mutation, you have arginine. If you look at the codon table, what you can see is that lysine and arginine actually lie in the same category. They both have positive charge. So the property of the amino acid hasn't changed. Now, if the property of the amino acid doesn't change, this means that the resulting folding action is also likely going to be conserved. So you fold the amino acid chain in the same way. However, if you have a non-conservative mutation, and we compare lysine, which sits in here with a positive charge, and threonine, this one has a polarity this time. So you can see that these two have different properties. Now, that is going to significantly change how your protein is going to fold. A polar amino acid, when it folds, it's going to be likely to stay on the outside of the protein because polar molecules are attracted to each other. So, an amino acid with a polar end is going to be attracted to water. Okay, next we have the chromosomal mutation. So we're past the gene mutation. We're going to look at a greater scale. So chromosomal mutation, there are two types. The first type is called numerical. This is when you have a change in the number of chromosomes inside a body. So a normal human set of chromosome, there will be 23 pairs of chromosomes. Number 1 to number 22 contains the gene for body functions. And then the 23rd pair is the pair that kind of determines your gender. Now you can see that in this person, when we do a scan, they do not have a normal number of chromosome. In the chromosome number 21, they actually have three instead of just a pair. So they have an extra chromosome 21, and we call this trisomy. Now, you don't have to memorize the symptom of Down syndrome. Everybody, sorry, I forgot to say that if you have three copies of chromosome 21, that means the person will have a Down syndrome. Now, why does this occur? Why does this happen in a person? So if you look on the left-hand side, this is a process of how sperms and how eggs are being produced in mammals. This is called meiosis. It's just a name for this type of cell division. So for meiosis, when you divide your cells up, the ultimate goal is to not have pairs of chromosome in those resulting egg cells or sperm cells. But the goal is to only have one of those copies so that when the eggs and the sperm fertilizes, then you have a complete set. So for example, a normal human cell, when we look at the pairs, one of them would have been from the egg, which is from the mother, and one of them would have been from your dad, which is from your father. So if this is a normal human cell, then on the right-hand side, this is a sperm cell's um, genetic scan. You can see that there is only one chromosome 1 and one chromosome 2. They are waiting for their Mrs. Right to pair up with so that they can have a complete set. Now, there is some sort of errors when it comes to producing these sperms and eggs. There's something called spindles, which is indicated by my cursor right here. Spindles are the fibers that pulls apart the chromosomes and split them up into two different cells so that you can have your egg cells with half the number of chromosomes. Now, when you're pulling your pairs of chromosomes apart to produce those eggs and sperms, sometimes the spindle snaps. And so instead of pulling one to each side, the spindle is actually pulling both the chromosome towards one end. And you can end up having the wrong number of chromosome inside the egg or inside the sperm. Now, this is more common when it comes to egg production instead of sperm production because human sperms are freshly made regularly every week, every month, just continuously until the person dies. However, it's different in the female's case. Females' eggs are actually pre-made and they're all made before they were born. So human ladies are born with all the eggs that they are ever going to use during their lifetime. And so They actually get these premature eggs and then they will mature them every single month. Now, here's the problem. If you have premature eggs made, it means that your spindles are there and they have been waiting for 20 or even 30 years before they are being used. It's just like a rubber band. If you have a rubber band sitting around for too long, you pull it, it snaps. So for ladies, if they have babies later in their lifetime, they're going to have very old spindles and they are unlikely to be able to perform the job normally. And so if we look at the rate of people having Down syndrome or Edward syndrome, which are babies with extra chromosomes, you can see that after the age of 35, the risk of carrying a Down syndrome baby or Edward syndrome baby actually grow exponentially. You don't have to memorize the process of meiosis, all right? All you have to know is the reason why you might get an extra chromosome, which is the weakening of the spindles over time. And that leads to the eggs having more chromosome than they should when they are dividing. So this is a trisomy 18. So Edward syndrome is when you have an extra chromosome 18, and you can see that the baby is more deformed than a Down syndrome baby. And I have a question for you. Which is worse, Down syndrome, Edward syndrome, or Pato syndrome? So one is a trisomy of the number 21 chromosome. The other one is trisomy 18, so chromosome number 18 having an extra copy. And then Pato syndrome is when you have a trisomy 13, which is an extra chromosome 13 right there. Now the answer is, oops, let's go back. The answer is Pato syndrome is the worst one, trisomy 13, because if you look at how the human chromosomes are lined up, the longest pair is called chromosome 1. The shortest pair is called chromosome 22. Now, if you think about it, having a trisomy 13, which means you have an extra copy of a chromosome that is relatively long compared to trisomy 18 and 21. If you have a longer chromosome, it means that you have more DNA bases, more information, and more genes sitting on that chromosome. As a result, more genes are going to be affected if you have a trisomy 13 instead of trisomy 21. Now, trisomy 13 is rarer than trisomy 21, and then trisomy 11 is even more rare. Now, I have never met anybody or seen cases where we have trisomy 1 or trisomy 2 or trisomy 3. Because if you look at the length of these chromosomes, they are super lengthy. They're almost twice the length of chromosome 13. And these babies will be so affected that they will never make it to birth. So they would die in their mother's womb and it will come across as a miscarriage. I think trisomy 11 would be kind of our max here. There are cases where we get maybe 3 to 6 months survival with trisomy 10 or trisomy 11, but rarely you're gonna find babies making it if they have a trisomy 9 or below. Okay, the last one would be chromosomal mutation, but structural. So the previous one we talked about having a different number, so those are numerical chromosomal mutation. Now we're going to talk about a structural chromosomal mutation. That is when you will have a deletion of an entire section from your chromosome, or you have an insertion of an extra section into your chromosome, or you could have inversion. So you're actually inverting two pieces of information in your chromosome, two sections. So you can see A, B, C, D. B and D has been inverted. They swapped places, and so you have A, D, C, B. And then last one, we have translocation. That is when you have a piece of chromosome from one chromosome being translocated to another chromosome. So imagine if this is chromosome 1 and this is chromosome 9. The W and X section on chromosome 9 has been translocated onto the end of chromosome 1. And so what ends up happening is that your chromosome 9 lost a section and your chromosome 1 gained a section that was originally from your chromosome number 9. So um, the effects of these kind of mutation is pretty big, but I can't give you two specific examples because they could happen on any chromosomes. All right, and that's about it. Don't forget about the test. The test is on, let's see. The test is on the 25th of January. So that would be Thursday next week. And yeah, that's it. Happy studying and learning. Bye.